CnH2ON. Okay. So the formula here is CnH2ON. So commonly they have given here as C.H2ON. So N here means, so N is number. Okay, it can be 1, 2, 3, 4 like that. So CH2O. That means you have to calculate like that. C2, suppose N is 2. So C2, H2, already 2 is there. So H2 into 2, H4O2. C2H4O2 is the compound formula. Okay, like that. Suppose glucose. Glucose is 6 carbon compound. So in glucose, how many carbons are there? 6 carbons. C6H12O6. This is the formula of the glucose. So like this we have to calculate. So here you can observe that you have glucose and fructose. So in the glucose, you have aldehyde group as a functional group. So first carbon, so the topmost one is having aldehyde group, CHO. Whereas the fructose molecule is having a keto functional group, that is C double bond O, which will be there on the second carbon. So it is highlighted. Okay. So other than that, we will have, every carbon will have H and OH molecules. So even based on their orientation also, the molecules will change. Okay, that we will discuss later. So this is the differentiation based on the functional groups, that is aldehyde and ketone. So the same thing here. So that is glucose, which is 6 carbon. So fructose is also 6 carbon, glucose is also 6 carbon. So you can see here. So other than that functional group, all the other factors are same. If you see, the formula is also same. So can you just count it? How many carbons are there in the glucose? 1, 2, 3, 4, 5, 6. In fructose, you have 6. C6 is common. Then H12. How many hydrogens are there in the glucose? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Even fructose, it is having 12. O also 6. Right? So that means the formula is same. But what is difference here? It is the functional group. So similarly, you can see in the glyceraldehyde and dihydroxyacetone. So it's a 3 carbon compound, glyceraldehyde. So C3O6, sorry, C3H6O3, right? According to formula. So I think if you calculate, you will get the same. Okay. So, but the only difference here is aldehyde and keto functional groups. Then these are the basic units. So the simple carbohydrate is this one, dihydroxyacetone and glyceraldehyde. So dihydroxyacetone is a 3 carbon keto carbohydrate, whereas glyceraldehyde is the 3 carbon aldehyde group containing carbohydrate. Now if you add one more carbon like that, you will get different compounds. So finally, you will get, suppose you have a glucose molecule. They are called as monosaccharides. Whatever this is, glucose, fructose, glyceraldehyde, mannose, sucrose, galactose, all these are monosaccharides. Mono means single, saccharide means sugar. So usually carbohydrates, we call them as saccharides. That means sugar molecules. When you add 2 sugar molecules or 2 monosaccharides, then they will become disaccharides. Okay. Then if you have 3 or more, then we call them oligosaccharides. If you have many such monosaccharides as a form of polymer, we call them as polysaccharides. So starch molecule is a form of polysaccharide. Okay. You see here, It's a simple representation. So you have 1, 2, 3, 4, 5, 6, 7, 8 carbohydrate molecules. But in real, you'll have many number, hundreds of molecules, monosaccharide molecules. They form a polymer, which is a starch molecule. It's a storage material, okay, storage carbohydrate. So when you digest, like when our digestive system, they digest this starch molecules, you will get a monosaccharides or monomers, which are sugars, okay, simple sugars. So what is their importance? So first thing is they are the source of energy. So brain cells and red blood cells, they require a lot of energy, especially brain cells. So this primarily they depend on the glucose molecule. Okay, carbohydrates, especially glucose. And this is on the like everyday basis. And when we want to store the energy, so that is glycogen. Glycogen is a stored form of energy in animals. Then, apart from this, apart from energy source, even these carbohydrates, they act as precursors or the starting material for the production of other organic compounds like fats, amino acids. So in the last class, I have told you about the lipids and fats. So if you see their metabolic pathways, you will see this glucose as the starting material or carbohydrates, some of the carbohydrates as a starting material. And then I already told you about the plasma membrane in the previous class. So where the lipids are present. So along with the lipids, you will also have carbohydrates. And apart from this, even in cellular functions like cell growth, adhesion of molecules, adhesion means attachment, then fertilization of egg and sperm. So in all these processes, you can see this carbohydrates. Then carbohydrate derivatives also, they also participate as drugs, especially in the cardiac glycosides, antibiotics. Okay, so like this, these carbohydrates are involved in various processes. They have very much importance in the biological process. And especially in the diabetes mellitus, we usually hear this term carbohydrates or sugar in simple terms. Because like as the whenever we eat food, our sugar levels, this carbohydrates, they enter into the blood and they will raise the blood sugar level, okay, after digestion and all these things. So that should be removed by the insulin, okay, which is produced by the pancreas. But when there is any impairment in insulin or when there is insensitivity, like our body, insulin will become insensitive. Like even though insulin is there, it cannot function on this sugar. So during like type 1 diabetes, type 2, all this. So during these processes, usually the sugar levels, they keep on raising, they won't come down. Okay, then that condition is called as diabetes mellitus. Okay, so these are the sources of the carbohydrates. So major sources of carbohydrates are bread, cereals, rice and pasta. So rice, as we eat as a like South Indians, usually they eat more rice. Okay, so that rice, they usually they raise. So for sugar patients, we usually say avoid rice or consume less amount of rice. So along that, bread, cereals, pasta, all these. And fruits. Fruits are the natural sugars. So they will be sweet. Okay. Although they are good, means for sugar patients, they should avoid some kind of fruits which are rich in sugar. Then meat, poultry, fish, eggs. So that means major amount of the quantity, more amount of sugars are present in these down part like bread, cereals, rice, pasta. Compared to that, fruits and vegetables, they have other vitamins and minerals along with the sugar. Then meat, poultry, fish, eggs, they usually contain more amount of protein, yogurt, milk, cheese, than the sugars. Then you can see fats, oils, and sweets. Sweets, obviously, we add sugar, right? So in fats and oils, you don't find much of the carbohydrates, right? So this is how the sources of the sugars. Now classification of carbohydrates. So I told you like monosaccharides, oligosaccharides, and polysaccharides. So monosaccharides, which are mono means single, which cannot be hydrolyzed further. That suppose that it is 3-carbon compound. Suppose you take glyceraldehyde. That is itself is a carbohydrate. You cannot further cleave that into another carbohydrate, right? So it cannot undergo further hydrolysis. But based on the number of carbons present, you have different types of monosaccharides. Like if you have 3 carbons, they are called trioses. Tri means 3. Like if you have again then 4 carbons, tetroses, 5 pentoses, 6 hexoses, 7 heptoses. Like that you will have. Then even in the trioses, tetroses, pentoses, hexoses, and heptoses, based on the functional groups, like if it is aldehyde group, then aldoses. If there is a keto group, then they are called ketoses. So there are aldoses and ketoses in all these 5 categories. So the examples of triose aldoses are, we call them aldotriose. Okay, glyceraldehyde. Then aldoketose, dihydroxyacetone. Aldotetrose, erythrose. Aldoketose, erythrulose. Aldopentose, ribose and xylose. Aldoketose, ribulose and xylulose. Aldohexose, glucose and galactose. Aldoketose, fructose. Aldoheptose, glucoheptose, and aldoketose, pseudoheptulose. So these are the examples. So the difference is the functional groups and the orientation of the OH and H molecules. So here you can see. Then you have glyceraldehyde. But in glyceraldehyde also, you will have D-glyceraldehyde, L-glyceraldehyde. So that is based on the orientation of the H and OH molecules on the second carbon. So here if you see, can you understand the difference? So the first carbon, it is having aldehyde group, that is CHO. In the second carbon, you have H and OH. H is on one side, OH is on another side. But only difference is D-glyceraldehyde having OH on the right hand side and H on the left hand side. Whereas in the L-glyceraldehyde, you have OH on the left hand side and H is on the right hand side. And on the third carbon, you have CH2OH. Okay, so how to identify? See, you cannot see them sugar. If you take a sugar, 1 spoon of sugar, can you observe that? What is the structure? What is the orientation? No, you cannot. So how actually you can observe whether it is a D glucose or L glucose? Of course, you can like you have to. There are some softwares you will go through the structure. All this that's fine. But here, when you take some sample of a sugar, you pass a plain polarized light, okay, plain polarized light onto that sample. So if that light shifts to right hand side or left hand side, okay, so based on that we can actually say whether it is D glucose or L glucose. If it is shifting to left side, L glucose. If it is shifting to right side, it is D glucose. Okay, that is the main difference. Then Now we will discuss about each sugar and its importance. Like glyceraldehyde and dihydroxyacetone, they are the intermediates of glycolysis. So glycolysis you all might have heard. So it's a basic first pathway we study. Okay. So where glucose is converted to pyruvate. And also, so in the glycolysis pathway, the center step, like in the like what you call bifurcation step, you will have this glyceraldehyde and dihydroxyacetone. Then we have, they are the precursors of the glycerol and lipid synthesis. So in the last class, I told you about the glycerol when I was taking the lipids class. So these are the precursors of the glycerol. And erythrose. So it's an intermediate of carbohydrate metabolism. Then ribose. Ribose. So in the nucleic acids, we have like DNA, RNA, right? You might have heard all this DNA, RNA. So in the DNA, it is deoxyribose. Okay. That means an oxygen molecule is removed from the ribose. So that is called deoxyribose. And in RNA, we'll have ribose. So this ribose, whether it is in RNA, DNA, deoxyribose, so that is nothing but this sugar molecule ribose. Okay, and also in some coenzymes, we find this ribose. Then glucose. So glucose is the main sugar of the body. So all our energy is dependent on the glucose. Then fructose. So fructose is converted to glucose and then it is utilized by the body. Okay. Then galactose. So galactose is mainly synthesized by the mammary gland. So this will make the lactose in the milk, in the breast milk. So whatever the lactose we see, that is a milk. So that is made up from the galactose. Then mannose. So it is mainly present in the glycoproteins and glycolipids. So in the bacterial cell wall, you will see this mannose. Okay. So in this way, like every carbohydrate, whether it is triose or tetrose or pentose, everything is participating in some or the other metabolic pathways. Then we have, these are all monosaccharides. Then oligosaccharides. So oligo means few in the Greek language. Few means how much we can consider, 2 to 10 monosaccharide molecules. And these monosaccharide molecules are linked together by glycosidic bond. There is a bond in between. So few means I told you 2 to 10. So if there are 2 monosaccharide molecules, you will have one name. There are 3, there are another name. Like that you have. So if there are 2 carbohydrates, then that is monosaccharide molecules, it's called disaccharide. If it is made up of 3, then trisaccharide. If it is made up of 4, tetrasaccharide. If it is made up of 5, it is pentasaccharide. So the disaccharide examples are maltose, lactose, sucrose. Trisaccharides are raffinose. Tetrasaccharides are stachyose. And pentasaccharides are verbascose. Okay. So di means 2, right? So maltose is made up of 2 glucose molecules, glucose and glucose. Lactose is made up of glucose and galactose. Sucrose is made up of glucose and fructose. Okay. Then raffinose is made up of glucose, fructose, galactose. Three carbohydrates. Stachyose is 2 galactose, 1 glucose, and 1 fructose molecule, like 4 monosaccharides. Then verbascose is made up of 3 galactose, 1 glucose, and 1 fructose. So 5 carbohydrate molecules. So like this, you will have different oligosaccharides and they are linked by glycosidic bonds. Then we have polysaccharides. So polysaccharides, as oligosaccharides are between 2 to 10 carbohydrates. So here if they are anything more than 10 monosaccharides units, then they are called as polysaccharides. So here again you have homopolysaccharides and heteropolysaccharides. So homopolysaccharide means all saccharides are of same type, all glucose, all galactose, like that. They are called homopolysaccharides. And again, there are two types, unbranched and branched. Unbranched means all are like single chain. So in this image, you can see. Branched means you will have branches from the main chain. Then heteropolysaccharides. So heteropolysaccharides means two monosaccharides. Like if you see one glucose, one galactose, one glucose, one galactose, like that, or glucose, galactose, mannose, like that. Like if you have different monomers present, then they are called heteropolysaccharides. Again, they are also can be branched or unbranched. So the examples of homopolysaccharides and heteropolysaccharides are homopolysaccharides, they are starch, glycogen, cellulose, insulin, dextrans, chitin, all this. Then heteropolysaccharides are agar and mucopolysaccharides. Okay, these are the examples. Then here we'll have isomers. So isomers means I told you like you will have same formula, but maybe the functional group or orientation of H and OH molecules they are called as isomers. Okay, they'll have same chemical formula but different structural formula. Like glucose and fructose, both having C6H12O6. But glucose is having aldehyde functional group, fructose is having keto functional group. Okay?
Excuse me, ma'am.
Yeah.
Ma'am, which chapter you are explaining?
So I told you right in the starting itself. Carbohydrates. In the biochemistry, you have carbohydrates, right?
Okay, ma'am, carbohydrates. Okay,
Yeah.
thanks, ma'am.
Yeah, yeah. Okay. So the glucose and fructose. Then stereoisomers. I told you about D and L. Okay. So D-glyceraldehyde and L-glyceraldehyde. So this is based on the formula is different, the structure is different. Both are having aldehyde group. But here the spatial configuration, we call it as spatial configuration. That means how these H and OH molecules are there in the space. Like one way it is like on the left hand side, in another it is right hand side. So based on that, you will have two types of um, same molecule, but it is represented in the two forms. So they are called as stereoisomers. Okay, D and L stereoisomers. Now you can see. So this is very important. Like not only for your exam point of view, like if you want to continue in this field, so you have to remember the differences between these all these structures. It's a very simple table, but you have to remember this. So I'll explain you. These are about aldehydes. Okay, aldehyde precursor is D-glyceraldehyde. These three carbons, okay, so just listen carefully and understand, okay. I'll just zoom it. So you have three carbons. If you have any doubts, you can stop me and ask, okay. Always the first carbon will have the functional group, that is aldehyde group. CHO is fixed. Okay, so always the first carbon is CHO, which is functional group aldehyde. And last carbon is CH2OH. Okay, this is also fixed. If you see in any of these carbohydrates in the below also, first carbon is CHO, last carbon is CH2OH. Just remember that. Now, you have 3 carbons here. So only the middle carbon will have the different functional groups, right? So in the middle carbon, you will have OH on the left hand side. So why they have not given arrow mark, that is okay. Means you can have right hand side, left hand side like that. That is another aspect. But just remember, okay, right hand side you have glyceraldehyde. Okay, now what you have to do is you take the same structure and keep at the below. Only thing is you have to add one more carbon at the second position. So as it is, take the D-glyceraldehyde group, add only this red color one, highlighted one, that is the newly added carbon. Okay. So now you have 4 carbons. C, 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 C. So first one is again CHO, last one is CH2OH, and third carbon is also same because you are taking it from the top, CHOH. Okay. Now you have to give the name erythros and threose. So erythros, you have the same structure. The OH group orientation is same as the precursor one. So OH is on the right side, H is on the left side. It is opposite in the threose. H is on the right side, OH is on the left side. Is it clear till here? Yes? Any doubts? Okay. Now, in the next stage, first you take erythros. Again, you take the same structure. Now you have to take erythros structure. Now you have 5 carbon. Okay. So this erythro structure is same. Only you have to add 1 carbon at the second position. And again follow the same rule. Ribose will have the orientation of OH as that of the erythros, whereas arabinose is having the reverse. So same thing in the xylose and lyxose. So same thing, you take same compound, make it into 2 structures. So orientation will differ. Like that you'll have D-allose, aldose, glucose, mannose, glucose, iodose, galactose, and talose. So therefore we give the name as aldotriose, aldotetrose, aldopentose, and aldohexose, where you have 6 carbon compounds. Is any doubts in this section, structures? Okay, so this is very important. Okay, you will have in competitive exams or whatever is, they will ask this question. They won't give you structure and all these things. You have to remember the formula. So I told you how the formula looks like, CnH2ON. So accordingly, you have to calculate how many carbons, how many hydrogens, how many oxygens. And based on the orientation also. First, you have to just completely remember this structure. Okay. So there is a term called asymmetric carbon atom. So asymmetric carbon atom is nothing but the carbon which is having 4 different functional groups attached to it. So in the first thing, suppose glyceraldehyde, this is the center carbon is the asymmetric carbon because on one side it is CHO, other side it is H, and other at the bottom it is CH2OH, and on the right hand side it is OH. So the same carbon is attached to 4 different functional groups. They are called as asymmetric carbon atoms. So now the number of possible stereoisomers depends on the asymmetric carbons. So like this is also important bit in competitive exams, not for your these annual exams also, but when you go for like PG or there like any entrance exams, they will have this number of stereoisomers possible. So the formula is 2 to the power of n. So now in the glyceraldehyde only one one asymmetric carbon is there, right? So 2 power 1. So you will have 2. So only 2 stereoisomers are possible, D and L. But if the number of N increases, the number of stereoisomers also increases. Okay. So like this here. Then all monosaccharides are derived from the glyceraldehyde, and the penultimate carbon atom is a reference carbon atom for naming the mirror images. So I told you, like, now you may get idea. Okay, suppose glyceraldehyde, already told you, like, OH is on the right hand side, H is on the left hand side. So we have given D and L. For glucose and large molecules, you will have more carbons. Then how will you give uh, L and D? Yeah. So that is based on the penultimate carbon. Penultimate means last one carbon, which is from the last, from the bottom. Like CH2OH is the last carbon. So one carbon before that, you have to see the orientation of H and OH on that carbon. Then you have to identify that as D and L, glucose or fructose or whatever it is. And then D and L isomers are called enantiomers. Enantiomers are nothing but the mirror images. Like when you see your hand in the mirror, so right hand side will appear as left, right? So here also these two sugars, they are called as mirror images. They can be superimposed. You can keep one above the other, but they are mirror images. That is the opposite orientation. And these sugars are naturally occurring sugars. That means you don't see L sugars mostly. Okay, only D sugars only you can see in the naturally in the nature. Okay, and they are called dextrorotatory. I told you, like, towards the right hand side or left hand side. So if you take a carbohydrate sample, pass a plain polarized light onto it, if the light rotates towards right side, then that is called dextrorotatory. If they rotate towards left side, then that is called levorotatory. So we call D sugars as dextrorotatory, L sugars as levorotatory. Okay. Hence, they are also called as dextrose. Okay. Then, so the same thing, dextrorotatory and levorotatory. You also represent that as plus and minus. In some isomers, you will have equal amount of dextrorotatory and levorotatory. So don't think that in nature they will be dextro or levo. So you will have a mixture of, so in the naturally occurring sugars, some will be dextrose, some will be levorotatory, and sometimes you'll have mixture of both. So when there is a mixture of equal mixture of both dextrorotatory and levorotatory, they are called racemic mixtures. Okay. So racemic mixture. So what happened in the chat in the fifth? Are you not able to follow like the syllabus or what?
Ma'am, whatever you are explaining, na ma'am, the topics
Yeah.
we are not having. We are having some other topics
So what are the topics you are having?
like monosaccharides, disaccharides, that is there. And after that, functions of carbohydrates we are having. Then metabolism.
Yeah,
Then
that
we
is
are
what
having
I'm saying, no.
glycolysis.
Yeah, before glycolysis, directly I cannot go to glycolysis, right?
Yes, ma'am.
First, I have to explain monosaccharides, disaccharides, and all these you should know. These are the basics of carbohydrates. Without
Yes,
knowing,
yes, ma'am.
yeah, syllabus they will give in a simplified way, but you have to read all these things. Without this, directly I will say D-glucose, L-glucose in the glycolysis. What you then again you will ask, what is D-glucose, madam? What is L-glucose? Right?
Yes, ma'am.
You have to tell what is D and what is L, no?
Yes, yes.
Yeah. So these are the basic concepts. Once I complete this, I will go to the pathways. Okay.
Okay, ma'am.
Yeah. So these epimers are nothing but the, like when sugars are different from one another, okay, they are called as epimers. So other than the reference carbons. So one carbon which is penultimate to that. So like this. So this D-glucose, D-mannose, D-galactose. Okay. So the reference molecule is the one carbon which is just above the last carbon. So in the third carbon, you have different orientation. So OH is on the right side and on the left side, like that you have. So if they are opposite side, then they are called as epimers. Okay. Then anomers. Anomers are nothing but, I will show you. Yeah. So we represent the glucose molecule in the form of three forms. Okay. So even in the pathways, while drawing your pathways, some will some will give you like Haworth formula, some will give you the open chain formula. Okay, some will add Haworth formula. Haworth formula is nothing but the ring structured formula. Open chain projection formula means like whatever the formula we are just reading. And another is Fisher's formula. So you can represent the carbohydrates in any of these three ways. But usually, like in your exams, when the question comes, not in the, okay, now they will just give what is the like glucose metabolism. You can write in the any of the formula like open chain or Haworth. But in some competitive exams, they will directly give the Haworth formula only. Then you should know which is first carbon, which is second carbon, right? So in the you have to remember the orientation of the H and OH in the open chain formula. So you see this glucose, take it as a reference. So CHO is in the first, OH is on the right hand side on the second carbon, OH, H, OH, OH. So this is the right hand side formulation. Okay. So in the Haworth formula, you see first carbon. Here, the O is already linked. Okay. So the oxygen on the first carbon is already involved in the ring formation. So now you don't see that. Okay. So you will see different orientation here. So H will be at the top, OH will be at the bottom. In the second carbon, H will be at the top, OH is at the bottom. So the form orientation is different, right? So that you have to remember. So no need of this hemiacetols and all. Um, okay, then you have mutarotation. Mutarotation is nothing but when D-glucose is crystallized at room temperature. So I told you, like whenever a solution is a light, polarized light is passed, it will either move to the right side or left side. That change in the rotation is called as mutarotation. Then we have disaccharides. So in the disaccharides, you have sucrose, maltose, isomaltose, and lactose. So sucrose is nothing but it has glucose and fructose. One molecule of glucose, one molecule of fructose, which is linked in the beta. So you have alpha D glucose, beta D fructose. These two are linked. So you usually name, how to name this is alpha D glucosyl, beta D fructoside. So this is the formula name of this sucrose. Okay. And this is the sweetening agent in the cane sugar. Okay. In the fruits, whatever the sugar we have, that is sucrose. So this hydrolysis of sugar, it will give the equimolar of glucose and fructose. So it is called as invert sugar. Okay. So why? Because it changes from the dextro rotation to levo rotation. And the enzyme which is cleaving this sucrose to form glucose and fructose is called as invertase or sucrase. That's why it is called as invert sugar. So the honey which is having invert sugar. Okay. So invert sugar is somewhat sweeter than the simple sucrose because it is changing from the dextro rotation to levo rotation. Then lactose. Lactose is present in the milk. Okay, main sugar. So here galactosyl beta 1,4 glucose. Both are like glucose and galactose. Here 1,4 linkage. So first carbon of the galactose is linked to fourth carbon of the glucose. Okay. Then, so in the digestion, if you see, usually this lactose, it enters into the small intestine where it has to break down into glucose and galactose. Okay. Suppose then it enters the large intestine. Okay. But in some people where there is a lactase enzyme, so a lactase enzyme which cleaves the lactose. So this lactase enzyme, if it is not present in some people, this lactose cannot be converted to glucose and galactose in the small intestine. So they directly enter into the large intestine. Okay. So in the large intestine, various bacteria, they will act on this lactose. After breaking of this lactose, they will produce some two carbon metabolites, carbon dioxide, hydrogen, all these, which will, when we drink water, so they will, along with the water and all these things, they will result in the diarrhea, dehydration, and bloating. Okay. So therefore, that condition is called lactose intolerance. So usually in small children, first we have to observe because they take breast milk, right? Usually some children, they will be colic. Colic means because they cry a lot because of gas formation. That is common in every children. But some children, they cry uncontrollably, unconsolably. Like that will be happen, like every day that will happen. So normal colic means one or the other day it can happen. But if this is continuously happening, so we have to check with the doctor. So this because for the lactose intolerance, if they have this, they cannot tolerate the lactose or milk. Not only in children but in also adults. And with age also, some people they cannot tolerate lactose. Okay. Then next is maltose. Maltose have two glucose components, which is having 1,4 linkage. Then isomaltose. So here 1,6 linkage is there. That is the difference. So I'm just skipping all these reactions. No need of this. Yeah. Lactulose. So lactulose is another disaccharide. So it is by galactose and fructose. Okay. So this lactulose is used as a medicine for the constipation. So these are the biomedical importance of glycosides. So you have these glycosides, they act as a drugs, cardiac glycosides. So digitoxin for cardiac insufficiency, so for the heart problems, and to stimulate cardiac muscle contraction, and they act as steroids. Okay, and they also Ouabain is acting as a sodium pump inhibitor. Okay. Then streptomycin, which is acting as an antibiotic. So in the tuberculosis, TB, they'll be given. Then fluoridazine, which is for renal damage, kidney problem, glycosuria. Okay, when more amount of glucose is secreted through urine, then this blocks the transport of sucrose across the mucosal cells of small intestine and renal tubular epithelium. So it will block the excretion of the sugar. Okay. Then amino sugars. Amino sugars are nothing but sugars along with the amine groups. So if you see in the amino acids, so that amine group when linked to the sugar, they are called as amino sugars. So like glucosamine, this is in the blood group substances heparin, hyaluronic acid. Then galactosamine, which is chondroitin sulfate of cartilage, bone and tendons. Then manosamine, which is constituent of glycoproteins. And N-acetyl glucosamine and N-acetyl galactosamine, these are present in the bacterial cell wall. Even mucopolysaccharides are present as they are the cell membrane antigens. So when the immune system, they identify these antigens from the bacteria and they will react. Okay. So I told you like deoxy sugars, deoxyribose, deoxyfructose. So mainly DNA is 2-deoxy-D-ribose. So at the second position, ribose is having oxygen, whereas DNA, deoxyribose is not having oxygen. So that is the difference between the RNA and DNA. Then homopolysaccharides. So we have starch, glycogen, cellulose, inulin, not insulin, inulin, dextrans, and chitin. So starch is the reserve, like cellulose in the form of, sorry, glycogen in the form of animal. So in the same way, starch is present in the plant, like potatoes, cereals like rice, wheat, and other food grains. They are composed of starch. So starch is having amylose and amylopectin. So this is how the structure of the amylose and amylopectin looks like. Amylopectin is a branched, amylose is unbranched. So amylose is made up of glucose subunits, unbranched long chain. They absorb water, okay, and they will form a gel, which is called amylopectin. So amylopectin is also having a glucose molecule, but it is in the branching point, which is alpha 1,6 linkage. Okay. I'm not going to explain you about tests. Okay. Then glycogen. So glycogen is the storage molecule in the animals, whereas starch, amylose, and amylopectin is a storage molecule in the plants. So glycogen is the carbohydrate reserve in animals. It is stored in the liver and muscle. So it increases during well-fed state. Like when we eat more, so after utilizing the enough calories, the excess amount will be stored in the liver and muscle. So whenever we fast, so that amount of glycogen will be utilized. So it is also having glucose molecules, but it is again branched. Okay, in each branch you will have 1,4 linkages, whereas at the branching points you will have 1,6 linkage. Glycogen is much branched than the amylopectin. So in the liver and muscle, so this glycogen, you have to remember this metabolism. So glycogen is converted to glucose 6-phosphate and then converted to glucose in the liver. So this glucose will be released into the blood. Okay, whenever we fast. Even the muscles, this glycogen is converted to glucose 6-phosphate. Again, that glucose is released. The next is cellulose. Cellulose is made up of glucose units combined with the beta 1,4 linkage. Okay, there are no branching point. They are hydrolyzed by the enzyme cellobiose, and this is present. This enzyme cellobiose is completely absent in the animal and human digestive system. That's why we cannot digest this cellulose. Okay. So this is how, like, it is a major constituent of the fiber. Fiber, undigested carbohydrate is nothing but the fiber. Okay, which cannot be digested. So this fiber can absorb 10 to 15 types of the weight of the water. Okay, that means it can, suppose it is 1 gram, it can absorb 15 grams of water, like that. Okay, 15 times. So 15 times its own weight in water. So this in the intestine. Okay, so this will increase the mobility. So it will, like, in people with a constipation, usually they have painful bowel movements, so that can have some bleeding or irritation and all these things. So when we take this lot, like, fiber food, so this fiber, which is undigested carbohydrate, it will protect the intestinal walls, and it will help in the bowel movement. So it will decrease the irritability or bleeding or like hard motion. Okay, bowel movement. So therefore, this fiber will increase the bowel motility. This will decrease the risk of the constipation, the cellulose. Okay, so it is given as a medicine for the constipation. And also it will reduce the LDL cholesterol levels. So this the bile salts and fibers. So fiber here is cellulose. Okay, they will reduce the cholesterol. They will take this cholesterol and they will excrete that cholesterol. Therefore, it decreases the serum cholesterol level. Okay. And they can go and bind to the toxic substances like carcinogens and eliminate them in the fecal matter. Thereby, it will reduce the chance of some cancers. And they also delay gastric emptying and can cause the sense of fullness. Okay, so that means some people it will be high, the blood glucose will be very high after eating the food, but it will suddenly drop after some time. So they will again feel hungry. It's again a medical condition, like frequently they have to eat something, but that cannot happen in this condition where we take fiber, more amount of fiber. It will give you the sense of fulfillness or sense of fullness. The next is inulin. It's a homoglycon. It is also glucose molecule. Okay, they are present in the bulbs and tubers like onion and garlic. They will help in renal clearance values and glomerular filtration rate. Then dextrans. So dextrans are also homopolymers. They are produced by microorganisms. So they will not go out of the vascular compartment. Hence, they are used for intravenous infusion of plasma. So this mainly in some condition, disease like hypovolemic shock. So in that condition, this dextran is given as a treatment. Okay. So dextrose, dextrin, and dextran. The terms look similar, but they are different. So D-glucose is called as dextrose. Okay. Then dextrin is partially digested starch, and dextran is the carbohydrate which is synthesized by the bacteria. So these three are different. Then next is chitin. So chitin is present in the exoskeletons of the insects, like in ants and all. We see this chitin. So it's composed of N-acetyl glucosamine with glycosidic 1,4-glycosidic linkages. Then we have heteropolysaccharides like agar and mucopolysaccharides. So agar is made up of agarose and agaropectin. So usually in microbiology experiments, so we prepare agar plates where we actually heat this agar and we will pour it and it will get solidified when it gets cooled. So it cannot be digested by bacteria. Hence we use this agar for culturing of the bacterial colonies. Then mucopolysaccharides are glycosaminoglycans. Okay, they are acidic sugar and amine sugar they form a polymer. So they have large amount of negative charge. So therefore, they extend in the solution and they repel from each other when we add water. Okay, so they will be slippery like mucus secretions. Okay, so this is similar to the synovial fluid. Okay, so they will be like slippery like mucus. How they will be slippery? They cannot mix with the water. So these are the glucosamine, glucuronic acid, and ururic acid. So these are the examples of glucosaminoglycans: hyaluronic acid, chondroitin sulfate, dermatin sulfate, keratin sulfate, heparin, their functions. Okay. Okay, we will continue in the next class anyway time is up. Okay, so this now we have finished the carbohydrate classification, their importance also whenever this thing. So only the pathways are left, even the lipids also pathways are left, and digestion also a little bit we have covered. Okay, next class we will try to cover the digestion and metabolism part. Okay, any doubts?
Okay, ma'am.
Okay,
Can we get the important questions, ma'am?
yeah, okay, next class I will discuss. Okay.
Okay, ma'am.
Okay,
How
okay.
many classes more we are having, ma'am?
I think two more classes, two more classes are there.
Hmm. Okay.
Two sets. Okay?
Okay,
Yeah,
ma'am.
yeah, yeah. Thank you.
Thank you, ma'am.